All right, welcome guys to section number 7. I think that's what it says. Yes. We're going to go over lectures 13 and 14. I'll try to do 15, 16 like back to back, but I have to go do something like in between. But yeah, I'm going to kind of not necessarily rush the slides because I actually want to do a good job, but also, I'm going to let the slides speak for themselves because I put a lot more text into this, so it's easier to follow if you want to just take it on your own run as well. I'll be explaining stuff briefly, like what's the most important parts of each slide and whatnot. So let's get into it. There's a lot of material to cover for this, and I don't have that much time. So lecture 13 is lipids and lipid metabolism. So basically, this is something I've taken from Sarah's words, these processes where we have breakdown of fatty acids, which is beta-oxidation, and then we have buildup of fatty acids, which is called biosynthesis. And so these processes are reverses of each other. However, they use different enzymes, so it's not completely reversible. Think of it like those irreversible steps of glycolysis and gluconeogenesis, right? Glycolysis, those 3 irreversible steps, you have to use an alternative enzyme to go the opposite direction. So it's a very similar case in this instance as well. Okay, moving on to the next slide. So we're going to move into the fatty acid oxidation. And I want to give a bigger picture before we go into like the details. So the first thing I want to talk about is what is a fatty acid, right? Or more so, like where did the fatty acids come from where we break them down? So I want to really clarify is that a triacylglycerol is essentially a glycerol right here, which is where we get this part of the name. It's the 3 carbons that forms our glycerol 3-phosphate or that kind of stuff. We'll talk about that in a little bit. And then our 3 fatty acids that are covalently linked to this glycerol. So it's elucidated here in this figure where we have the glycerol, it's a 3-carbon backbone, and then the 3 fatty acid chains that come off of it and branch into it. So that's basically what a triacylglyceride is. Now we got to talk about what each part breaks down into. So I want to talk about specifically first this glycerol. So we're basically going to cleave these bonds right here. We're going to pluck apart the 3 fatty acid chains from the glycerol, and we're going to end up forming a free glycerol right here. So what ends up happening is this glycerol will be converted into glyceraldehyde 3-phosphate, which we all know it's part of glycolysis. It's after aldolase and the triosisomerase enzymes converge and form this G3P. So first thing that happens is we have our glycerol through glycerol kinase. Obviously, a kinase means that's phosphating, or it's going to take a phosphate group from ATP and add it to the glycerol. So it's going to add it on the glycerol 3-phosphate. Now I want to emphasize that this enzyme will always form the L-glycerol 3-phosphate, and that isn't the same as our D-glyceraldehyde 3-phosphate. We have to do some interconversions essentially. So basically the first step is we activate our glycerol with ATP and we form this L-glycerol 3-phosphate. We can't necessarily just oxidize it, because then if we were to convert this OH into the carbonyl group, this carbonyl group, in order to oxidize this, we're going to be forming L-glyceraldehyde 3-phosphate. But in this case, we see that it's a D-glyceraldehyde 3-phosphate. So we have to do this interconversion, which is step 2. So step 2 is a redox. So notice how the OH turns into a carbonyl double bond. So right here, in this second part, we're going to form this carbonyl double bond. What that ends up doing is it converts the C into an sp2 configuration. All it is it just makes it planar. And then when we introduce this triose phosphate isomerase enzyme, it's going to convert it to D-glyceraldehyde 3-phosphate. So just something to note. And I think that's mainly everything for this slide that you should know. Okay, so then the next step is activating fatty acids. So now that was the glycerol. We know that it goes into glycolysis through G3P. We know that. Now, what about the fatty acid chains that we just plucked off? Well, those are going to undergo its own separate pathway, which is beta-oxidation. So just want to emphasize first, before we even begin beta-oxidation, we have to do activation, whiwhich again, it's very similar to Krebs cycle. It uses the CoA enzyme. So we know CoA forms that thioester bond, which we see here. This is the activated fatty acid. This is the end of it, not the whole thing. So the R is obviously going to be the chain, but this is going to be the end of it with that CoA thioester bond. And this is now the activated fatty acid. And the enzyme that does this is called fatty acyl-CoA synthetase. And synthetase should be a giveaway that it uses ATP because the TACE. And as you can see here in this mechanism, it does end up using the ATP. And what drives this reaction forward in the spontaneous direction, again, is that pyrophosphate dissociating into 2 inorganic phosphate molecules. So it drives the reaction forward because you're removing the products right away. And then, yeah, that's mainly for this slide. Sorry, I'm going a little bit too fast. We got a lot of material to cover. So the acyl-carnitine shuttle. So essentially, we're in the cytoplasm, right? Pretend we're taking up triacylglycerides from the blood and it's going into the cell, right? It goes into the cytoplasm, but now we need to get the fatty acid chains that we have just activated from the cytosol into the matrix, because beta oxidation only occurs in the matrix, or in the mitochondrial matrix. that is, you have to know this, is that beta oxidation does not occur in the cytosol, it occurs in the mitochondrial matrix. And so this is what this shuttle is. It's a new one. It's called the acylcarnitine shuttle. And what we use is this carnitine, like protein. Pretty sure it's a protein, but besides the point, we use this molecule where it's going to basically convert with the thioester bond, so it's going to like interchange, and the carnitine is going to attach itself to the fatty acid, or the fatty acid is going to attach itself to carnitine. Doesn't really matter, but this is what ends up happening. And when this molecule forms, the fatty acid now has the ability to go from the cytosol through the intermembrane space and then through the matrix. So right here, as we can see, the carnitine can obviously freely diffuse the outer mitochondrial membrane because we know it's permeable. So it's going to permeate through here. And then from there, it's going to move on through this transporter that is only permeable to molecules that have carnitine attached to it. It'll allow the fatty acid to go in here. And then eventually, we're going to have another CoA enzyme just do the interchange again. And now our activated fatty acid is inside the matrix. And then the carnitine is able to go back into the cytosol and repeat this loop. And the enzymes that catalyze these carnitine substitutions is called the carnitine acyltransferase 1 and carnitine acyltransferase 2. These are they are distinct. So you do have to know that number 1 is the one that converts it initially in the cytosol. And then number 2 is the one that basically removes it from the fatty acid chain once we're inside the mitochondrial matrix. And something I really, really want to emphasize is that the formation of acylcarnitine is the rate limiting step in beta oxidation. So if we know beta oxidation is a breakdown of fatty acids, the rate limiting step is the shuttle between the two. And what is it inhibited by? If it's a regulation, or if it's a committing step, if it's a rate limiting step, that means it's going to be highly regulated. So the regulatory molecule is malonyl-CoA. And this will become important, but malonyl-CoA is essentially a precursor for fatty acid synthesis, right? So we have acetyl-CoAs and we're building them up into fatty acid chains. And one of the precursors, the starting molecules, is malonyl-CoA. So it makes sense that if we want fatty acid synthesis, which we'll talk about in depth, I'm just explain this conceptually. If we want fatty acid synthesis to occur, we don't want the opposite direction to keep going, right? We don't want beta oxidation to be activated. So malonyl-CoA is going to inhibit that other pathway so that fatty acid synthesis could be the dominant pathway. This should be very similar to glycolysis and gluconeogenesis. Moving on to the next part. So I think this is just a reiteration of exactly what I just said, just much more in detail, like where you start, where you end, and specific enzymes that are able to do so. So I'll just skip this slide. So now moving on to the actual beta oxidation, right? We have our fatty acid inside. It's activated. How do we break it down, right? So that's what beta oxidation of the acyl chain is going to do. So basically, we're going to clip our fatty acid molecules into smaller components, 2 carbon molecules, like components, monomers, I guess you can call them. They're essentially acetyl-CoAs, as a giveaway. And then they release energy in form of reducing equivalents. So they will form NADH and FADH2 and also release acetyl-CoA, which we know will go through the Krebs cycle. But there's a lot of energy that we can harness from this. So that's what I want to emphasize is like we generate the reduced cofactors and we also generate the acetyl-CoAs, which again go through TCA cycle. So just want to emphasize this. And something I really want to point out is that this 8-acetyl-CoA is from 16 carbon fatty acid chain. If it's a different one, like for example, if it's 18 carbon chain, right? Instead of having 16 here, if it was 18, then it would be 10 because we have to divide it by 2. So hopefully that makes sense. Just want to emphasize. And then yeah, electron transfer chain. So this kind of briefly summarizes where the products of beta oxidation go, the energy cofactors, and ultimately how we generate the ATP in general. So let's move on to this. So the 4 steps of beta oxidation, this is like the bread and butter of what we're trying to talk about in this lecture. And so what I want to emphasize is that we have a palmitoyl-CoA, which is a 16 carbon fatty acid chain, right? But we're only going to talk about just breaking one of these 2 carbons apart, right? We're going from 16 carbons right here on the top to the 14 on the bottom. So for the first part, we basically have this palmitoyl-CoA. It's going to be basically oxidized, my apologies. So we have this palmitoyl-CoA, and it's going to be oxidized into whatever this delta 2 enoyl-CoA. Basically, I just call it trans-enoyl-CoA. Like that's basically how I do it, or how I would pronounce the product at least. And the enzyme that does this is called acyl-CoA dehydrogenase. And what it does is it's going to take an H from the alpha carbon and an H from the beta carbon and basically pluck them off. And what's going to end up happening is we form that double bond. And that's technically an oxidation of our fatty acid chain. And if we're oxidizing the fatty acid chain, we have to reduce something. And that's where we generate the reduced cofactor. So the first step is a redox reaction. So something to keep in mind. And then that's step 1. So step 2 is called hydration, where we essentially we form that double bond between the alpha and the beta carbon. And this double bond, we can hydrate it with water. It's like the first reaction you learn in Orgo 1. It's very simple. You just add a water. It's going to add an OH to one of them and then an H to the other. Specifically, it's going to add the OH to the beta carbon and an H to the to the alpha carbon. And so that's our hydration step. There's nothing fancy about it. We just add water. Then the third step is going to be our second oxidation, right? Where we see here our water that we just added, the OH that got attached to the beta carbon, is going to be oxidized into a carbonyl and into a ketone group. And so when that gets oxidized, we have to reduce something in its place, right? So that's going to be our NAD plus that gets reduced to NADH. And that's our second set of reduced cofactors. As you can see, NADH, it's going to be passed on into ATC respiratory chain to generate ATP. And you guys already are familiar with that. So that's where these 2 electron carriers come from. And then that's the third step, right? Now the last step is to actually like cleave this 2 carbon molecule off of the rest of the chain, right? So we're going to use this acyl-CoA acetyltransferase, which is just, you can just write simply thiolase. And this enzyme, what it's going to do is it's going to come between the alpha and the beta carbon. And what it's going to do is basically just cleave it by addition of the coenzyme A and forming this thioester bond here. Now, I want to make this a point is that we have our alpha carbon and then the carbon that's the carbonyl that's attached directly to this S, right? It's going to be the bond between the alpha and the beta that cleaves off, and we form our acetyl-CoA, right? And that our acetyl-CoA is our 2 carbon monomer, 2 carbon monomer for the fatty acids. I guess is how you can put it. But I just want to emphasize that each time you undergo this loop, right, you're going to end up generating your acetyl-CoA, 1 NADH, and 1 FADH2. Just every single time you go through this loop. Okay, so the next step is comparing energy yields. So beta oxidation is more energy efficient than glucose metabolism, and we'll get into why our body doesn't just use fat instead of glucose. So we'll talk about that in a little bit. But just some like mathematics, you guys should be very, very familiar on your exam. And this is in my year. This was in the year that I was TAing last year. Every time there has been a calculation question, which you can either love it or hate it, but I'm letting you know ahead of time, like you should definitely go through this very intensely and slowly to understand where all these numbers come from. So glucose, it yields 5.33 ATPs per carbon max. And that has to do with 32 ATPs produced per 6 carbons in the glucose molecule itself when you start it. And palmitic acid, which is just the 16 carbon fatty acid chain, ends up generating 6.625 ATP per carbon. And that's because we produce 106 ATP from 16 carbons in the palmitate. And so you may ask, well, where's 106? Run through the numbers. So we have the 7 cofactors of FADH2, the 7 cofactors of NADH. We end up generating 8 acetyl-CoAs because the last step, remember, generates the 2 acetyl-CoAs. It does not generate another chain. You have to go through the process 8 times. You only go through it 7 times. Hopefully that makes sense. If that is confusing, just do the beta oxidation, like draw it on an iPad, like go through each step, cutting off 2 carbons at a time until you reach the last step, and then it'll become super clear where it's like, oh, well, yeah, I generate 2 acetyl-CoAs, and this just makes sense why it's only 7 steps, not 8. So if that is confusing, just go through the beta oxidation multiple times until you end up with all acetyl-CoAs, and this will become extremely clear. But before I move forward, so we have our 8 acetyl-CoAs, right? Remember, through Krebs cycle, for each acetyl-CoA, we're going to end up generating 1 FADH2, 3 NADHs, and 1 ATP, right? So we multiply all those numbers by 8, and we end up forming 8 FADH2s, 24 NADHs, 8 ATPs. And then we have to do the interconversions, add the net stuff, and it's pretty self-explanatory here. Also, on the exam, you will not be able to use calculators, so definitely like get good with this mental math. But yeah, as you can see, we have 15 FADHs too, that's net, 31 net NADHs, and net 8 ATP. We do all the conversions. We convert it to 108 ATP minus 2 ATP, and that forms our 106. Now, where does the minus 2 come from? Remember in that first step, I didn't really emphasize this as much as I would have liked, but that first step, we end up activating using an ATP. But we end up using 2 equivalents because we form this pyrophosphate that breaks down. So that's where those 2 ATPs come from. Yeah. So basically, just to reiterate, I just want to reiterate that the first step, we basically take this total amount of ATP that we have and we subtract 2 ATP. And this is because we use 2 equivalents. We go from ATP from that first step to AMP, not ATP, AMP. And that means that we use technically 2 equivalents, so that's why we subtract by 2. If you want to see it, I'll show you right here. As you can see, we have on this formula, ATP is on this part right here, and then it turns into AMP. So we can consider it as 2 equivalents. So there's a reason why we don't just use fat energy all the time, even though it's probably more energetically efficient, or we can maximize more per palmitic acid or whatever. There's a reason why. And the reason is because glucose can be stored as a hydrophilic glycogen and it can compact really nicely, whereas fats are hydrophobic and are stored in separate specialized cells. And so they're not necessarily in it's not ubiquitous in all cell types. It's only going to be in adipose tissue, adipose cells. And so the fats, they're very much not able to compact as much because those fatty acid chains are such long chains and they can't really bend, especially when we start introducing double bonds and triple bonds, which, well, more so just double bonds. But I don't want to get too far ahead of myself. But the fatty acid chains can't necessarily fit in very neatly, right? Not as neatly as glycogen would. So that's one of the reasons why we use glucose, is because we can store more in a compact area, and it's ubiquitous across all cell types, whereas fats are only really found in adipose tissue cells, if that makes sense. So now I want to move on to what happens if we have a double bond, right? Saturated fatty acids are essentially just all single bonds, no sign of a double bond in the chain at all. Now, unsaturated means that we can either have 1, 2, 3, 4, however many double bonds in the fatty acid. So whenever we see unsaturated, we know, okay, double bond is there, right? And we don't know if it's cis or trans. That will come later, and we'll talk about that in a little bit. But so far, we've assumed that beta oxidation occurs with just saturated fatty acids, these single carbon, or these no double bond fatty acid chains. And so how do we deal with the double bonds, right? So this is just examples of what that looks like. And so there's either two scenarios, right? So what ends up happening when we have to oxidize unsaturated fatty acids, and there's two issues at hand. There could be an issue where we only have one double bond, right, in this fatty acid. And then the second issue is, what if we have multiple double bonds, right? So what ends up happening is, well, we have to first go through this. And so for the first one, we have a monounsaturated fatty acid with one cis double bond. And so what I want to emphasize is that as we're cutting our 2 carbon units and forming our acetyl-CoAs and making this chain shorter, we're going to come across a cis double bond. And so whenever we have a cis double bond, we're going to be using this enzyme called enoyl-CoA isomerase. And you don't have to worry about the deltas. Just know that's an enoyl-CoA isomerase. And so what you're going to end up doing is this enzyme is going to convert that cis bond. And I want to emphasize that that cis bond is found between the beta and the gamma carbons here. So we have alpha, beta, gamma. And it's going to shift the double bond from beta, gamma to a trans double bond from alpha, beta, as you can see here. So all you got to do is just label the alpha carbon that's adjacent to this carbonyl carbon. That's alpha, beta, gamma. And then this one is alpha, beta, gamma. So you can see that the placement of that double bond changes. And then you can undergo beta oxidation. And that's the solution, is that we stop at this trans double bond. And we know from beta oxidation right here that this double bond step right here is our trans enoyl-CoA, which is our just basically trans double bond fatty acid here. And so we start from this step now, instead of starting from this what's called palmityl-CoA, starting from the just single bond CC between the alpha and beta. We just skip this step, this oxidation reduction reaction, and we move on to the second part. And so we basically lose a cofactor whenever we have this double bond in the cis double bond inside of the fatty acid, right? Now, what about the second problem, right? Where we have multiple double bonds, right? So in this case, we're going to use a different enzyme. We're going to add an extra enzyme to the previous one. So we're going to be using this thing called 2,4-dienoyl-CoA reductase. What it's going to do is it's going to basically undergo an oxidation reduction reaction where we're going to have a trans double bond between alpha, beta, but we're going to have a cis double bond between gamma and delta. There's no double bond between beta and gamma, as you can see here. That is a single bond. But what ends up happening is when we reach the stage where the cis bond is basically going to be the next unit that's going to form, what we're going to do is introduce this enzyme, and it's going to basically collapse these 2 double bonds together. You can think of it like this. They collapse the 2 double bonds, and now they're located between the beta, gamma. So right here is alpha, beta, gamma, delta. So as you can see, we take the 2 double bonds, we basically mesh them together, but it forms our trans double bond that we need. And then the last step, or actually this forms the cis, but anyways, last step is that the double bond has to be between the alpha and the beta, as you can see here, in order for beta oxidation to continue. In this case, it's between beta and gamma, so that's not in the correct position. So that's where enoyl-CoA isomerase comes in and is very helpful, right? So in this enoyl-CoA isomerase is going to convert that beta gamma double bond to the alpha beta now it's in the correct position, and we can continue beta oxidation. And that's the same thing here. It doesn't necessarily have to be cis. It's just the fact that the double bond is between beta and gamma. So right here's alpha, right here is beta, and then right here is gamma. So we have to always have the double bond on the alpha beta carbon in order to continue with beta oxidation. As you can see here, we have alpha, beta, and then gamma. So if the double bond's here, we can't continue. We can't proceed with it. We have to convert it into the alpha beta right here, and we use the enoyl-CoA isomerase enzyme to do so. So that's hopefully the two solutions. Watch it back if it was a little bit confusing. Hopefully that clarified a lot about this. The next thing is, what if we have an odd number of fatty acids, right? This is like the final problem that we could run into. So what if we have an odd number, right? What if we break down all the chains and we form instead of forming 2 acetyl-CoAs at the last step, we form our acetyl-CoA and then a 3-carbon, this propanyl-CoA molecule, right? We can't necessarily just break it down so that there's only 1 carbon and then our next acetyl-CoA. We leave this intact. And so our body actually has this enzyme called propanyl-CoA carboxylase. And something I want to know is that, again, it uses biotin, and we know biotin uses ATP. So remember this coenzyme and these cofactors, these cofactors, ATP, is associated with biotin, this coenzyme. So what's going to end up happening is we're going to attach another carbon. So from a 3-carbon to a 4-carbon, we're going to convert this 3-carbon to a 4-carbon now, like fatty acid molecule. So it's going to go from propanyl-CoA to methylmalonyl-CoA, right? Now what ends up happening is we have to use this methylmalonyl-CoA epimerase, so which means we change the configuration of it, right? This is a D configuration. We're going to change it to the L. And so all it's going to do is just shift the carbon dioxide and the CoA bond right here and basically convert it. So now we end up forming our L-methylmalonyl-CoA. And the reason why we need this is because we want our CoA enzyme to be near our 3rd carbon up here. So as you can see on this part right here in the D-methylmalonyl-CoA, our CoA part of the substrate of this molecule is super far away from our 1st carbon right here on the top one. So we have to do the epimerase step first, and that gets it geometrically or spatially closer. And then from there, we can do the conversion using this methylmalonyl-CoA mutase. And it does use coenzyme B12. And this is something that you do have to memorize, that it uses this coenzyme. Because they could ask, like, what if you have a vitamin B12 deficiency? What pathways could be altered? This is the exact pathway. We would have an extremely hard time being able to break down or use our 3-carbon fatty acids. So the final result is that we form succinyl-CoA. Succinyl-CoA is part of the Krebs cycle, so that's exactly where it feeds into. Now, this is that I want to emphasize. It is a anaplerotic reaction. So remember how we said, and this is not to get too abstract, remember how we said in animal cells, we can't necessarily live off of triacylglycerides or acetyl-CoAs or fatty acids because we don't have the ability to replenish the Krebs cycle intermediates. There's no ability for us to use acetyl-CoA as an anaplerotic reaction. Well, in this case, it's not necessarily that we're using acetyl-CoA as an acetyl-CoA. We're using a 3-carbon CoA, and it's very similar. But what's going to end up happening is it gets converted into succinyl-CoA, and this now can go into the Krebs cycle and replenish the intermediates. So for some instance, you can think of this as an anaplerotic reaction right here. So that's one way to connect it to Krebs cycle again. I think that's everything I wanted to say here. So moving on to the next one. So now the next thing is, this is I'll give you the context behind this in terms of using ketone bodies and ketogenesis. So for whenever we don't have glucose available, we end up relying on fatty acids. They take a lot more processing power, it takes more time, and it's not the most efficient as glucose, but our body sometimes will rely on this, and that's why like people will do intermittent fasting, they will do all this stuff to promote fat loss because they want to break down the fat stores in their body. And so the way we break down these fat stores and utilize it as energy is through these ketone bodies and ketogenesis. So ketone bodies are derived from acetyl-CoA, the broken down monomers of fatty acids, and they're made during starvation, and they're used as a way to fuel the body whenever we're going through times of starvation. And so ketogenesis occurs basically only in the liver, and KBs are released or exported from the liver into the bloodstream to be used by vital organs. Now, it's a whole complex pathway. We will break it down. And so during starvation, I just want to give the context, gluconeogenesis depletes TCA intermediates. So our TCA cycle is basically doesn't work anymore. TCA slows and acetyl-CoA builds up. Now, the acetyl-CoA can be turned into acetoacetate. Acetoacetate is just 2 acetyl-CoAs mashed together, and then you get rid of the CoA right here. As you can see, there's the first acetyl-CoA, and there's the second one, and we got rid of the CoA part of it. Then KBs, these ketone bodies, are sent to the brain, muscles, and kidneys, specifically the brain, because we know that at first the brain loves to use glucose. It will only use glucose. It'll draw all the glucose from the body to it. But once all the glucose is gone, it will draw the ketone bodies, and our brain can actually survive off of that for a little bit before we undergo acidosis. But it's besides the point. All right, so now let's go into the ketogenesis process, right? It's like I mentioned, we're going to form our acetoacetate using 2 acetyl-CoAs and meshing them together, right? So acetyl-CoA, 2 of them are going to come together through the thiolase enzyme, because remember, thiolase is kind of reversible, so we end up forming this acetyl-CoA. And then the next thing that's going to happen is we're going to form our HMG-CoA synthase. And remember this enzyme because it's also used for another process as well. And so the HMG-CoA synthase will basically convert our acetoacetate-CoA into 3-hydroxyl-3-methyl-glutaril-CoA, HMG-CoA. The only conversion is that we're adding another acetyl-CoA right here. So you can see another 2 carbons. We have 4, 1, 2, 3, 4, and we're adding another 2 to form a 6-carbon molecule. And so we build up this like complex molecule, 6 carbons, and then we use HMG-CoA lyase. And that's what this substrate is called, HMG-CoA. And so what we're going to do is take out 2 of these acetyl-CoAs at the very bottom, and that's going to be removed. Now, we now have formed our acetoacetate, where there's no CoA found in this product at all. And as you can see how we're able to do so with with the initial steps. And then what ends up happening is acetoacetate can spontaneously break down into acetone, and we know acetone is very toxic to our blood, but it does result in toxicity in the blood in terms of acidification, all that stuff. So it's bad for our body to solely rely on acetoacetate. Now, secondly, it can also be converted into beta-hydroxybutyrate, which is just another 4-carbon molecule, but instead of it being a ketone group on the beta carbon, because this is our carbon 1, alpha carbon, beta carbon, it's going to be a hydroxy group, OH. So that's why we name it beta-hydroxy and then butyrate, because it's a 4-carbon carboxyl and terminus at the end, a carboxyl group at the end of it. So hopefully that naming makes sense, but this is the whole process of ketogenesis. Definitely know all the steps, know all the enzymes, know all the cofactors, basically everything that happens, because this is a process that's very important to our body when we're starving. So basically, the acetoacetate can be transferred through the blood, and same with the beta-hydroxybutyrate. So it can be basically transported throughout the body, and same with acetone as well. It can go in our blood, and so these 3 ketone bodies can actually be basically will go into the blood. They will travel to the vital tissues that they need to go to, so like the brain, for example. And once they get to the brain, they will be converted back into acetyl-CoA, so we can actually generate ATP when we need it. So the way it does so is let's say we have a beta-hydroxybutyrate in the blood, and it gets to the brain. And then once it's inside the neurons and all the supporting cells, all that stuff, we're going to have this beta-hydroxybutyrate dehydrogenase molecule basically conduct this oxidation reduction reaction, where we take our beta-hydroxybutyrate and we're going to oxidize it to acetoacetate. And this acetoacetate, what's going to end up happening is the carboxyl group, with acetoacetate, what ends up happening next is we're going to activate it into acetoacetyl-CoA. So we're going to basically turn into like a 4-carbon acetyl-CoA, if that makes sense. I'm not trying to confuse you even more. But the way it does so, and does this process, the second step, is through this enzyme called beta-ketoacyl-CoA transferase, where we're going to basically take our succinyl-CoA, a free one from the Krebs cycle, and we're going to convert it into succinate. It's almost like we're going through an alternative step of the Krebs cycle, but it still has like the same substrate and products. And it's gonna take that CoA off of the succinyl and attach it to this acetoacetate to form our acetyl-CoA. And sorry, that's like a mouthful. And then in our final step, what ends up happening is we use this thiolase to now cleave it and do the final step of beta-oxidation. That's essentially it. And now we have 2 free acetyl-CoAs that can be used in the Krebs cycle and basically keep the energy flow going for however long. So yeah. And so that's basically ketone bodies, beta-oxidation, and that covers basically like what the cell does in terms of using fatty acids, lipids as the main source of energy. So now, let's say we have a lot of energy, right? Remember when I talked about having high glucose, right? Having very high glucose, but also having high energy. And let's say that, glycogenesis, and there's a lot of glycogen as well, right? Well, if there's a lot of glycogen, a lot of glucose, and a lot of energy, what do, right? How do we convert it? And so the thing that ends up happening is if we have high energy is that we will activate this fatty acid biosynthesis pathway. So if we can't build up any more glucose, it's full. What we end up doing is taking that extra energy and converting it into fatty acids now. We take the acetyl-CoAs and now we're forcing them together. Remember, we can reap a lot of energy, but in the same way, we can store a lot of energy in these fatty acids. That's exactly why if we're in a calorie surplus, we have a lot of energy, we have a lot of glucose, we end up forming fat, and it forms in our adipose tissue. That's exactly why. So the first step that we have to do is basically have this acetyl group shuttle. And so the acetyl-CoA must be transported from the mitochondria to the cytosol where fatty acid synthesis occurs. Again, fatty acid synthesis in the cytosol, beta-oxidation in the mitochondrial matrix. Know that they are spatially separated. And something I want to mention is that acetyl-CoA is not exported directly. So the acetyl-CoA has to combine with oxaloacetate and form citrate. That is the very first step of Krebs cycle. And when it forms a citrate, then it can undergo this shuttle and essentially leave the cell. Now the citrate, through citrate lyase in the cytosol, right here on this diagram, it will basically break apart into oxaloacetate and also acetyl-CoA. And this is almost like the malate aspartate shuttle. Not quite, but it's very similar, or the malate can now go back, right? And then, yeah, we end up forming all this stuff. But that's what I want to emphasize is this top part right here. So I have this next diagram is that it separates it. So you start on this part of this diagram and then you end off on here. And something I want to emphasize also is that this step will also generate ADH as well that we can use to build up fatty acid synthesis. As you can see, generate cytosolic NADPH. And again, this process uses 2 ATPs per acetyl-CoA. So as you can see, there's 1 ATP used here and another one used over here. So that's where the 2 ATPs come from. So the top half is the phase of interest in terms of why we want this for fatty acid synthesis. And then the bottom half is like the reset phase where we have redox reactions. We have these transporters, these substrates that are able to go through the transporters and reforming the oxaloacetate for the interest part of the shuttle, the top part, right? So in order to have this top part go, we need to regenerate and reset the oxaloacetate that's out in the cytosol. So that's what this bottom half is for. Yes, you should know all like the enzymes, know all the specific substrates, which transporters. You should definitely know this like in depth. And again, if this is overwhelming because there's a lot of pathways, draw all of the pathways into one iPad, like just unlimited like space, because I know there's like a free Apple app or something like that. If you are on GoodNotes like me, there's a free Apple app that allows you to use as much space as you want. Put every pathway on there and note their convergence and divergence. Like note where they overlap and where they don't, because that's going to really help you set in stone where these pathways overlap, which ones are the same, which ones are different. How can some molecule like navigate its way and into a certain pathway and then go to another as well, like these alternative pathways. That is the best way to study for this exam, in my opinion. So moving on to the next one, the beginnings of fatty acid synthesis. So let's say we just transported the fatty acid outside of the cell, right? So the fatty acid is synthesized from malonyl-CoA. Remember, malonyl-CoA is a 3-carbon molecule. So it's not acetyl-CoA that forms our fatty acid synthesis. We don't just start adding them like Lego bricks, the 2 carbons at a time. We have to form a 3-carbon first and then add the 2. And this will be clarifying a little bit, but I just want to talk about the first step. Our acetyl-CoA carboxylase is the enzyme that catalyzes the first step. So basically what it's going to do is it's going to add a carbon dioxide using biotin, all that stuff. We talked about this already. When we use a carboxylase, we use biotin and bicarbonate or the CO2. And we're going to add this carbon dioxide onto a acetyl-CoA. And that forms our malonyl-CoA enzyme. And again, this step is the committed step. So remember, beta-oxidation was the transport of the fatty acid, the activated one, from the cytosol to the matrix. That was a committed step for beta-oxidation. This one is the formation of malonyl-CoA, to add that extra carbon onto acetyl-CoA. That is the committed step. This is the rate limiting step. This is where it's going to be highly regulated. And then the regulation, again, it's inhibited by glucagon. This makes sense. If we have low glucose levels, we want to use our energy reserves not to build up fatty acids, but to build up the glucose. That's our primary energy source. So if we have glucagon secreted, our body is telling us, hey, we need the glucose to be generated instead of the fatty acid chains. So that's exactly what's going to end up happening. It's going to favor the gluconeogenesis pathway. And same thing with epinephrine as well. And then the next one is palmitoyl-CoA, is a downstream product which will actually inhibit this. If we have a lot of product built up, we don't want to build up anymore. So it's almost like product feedback inhibition, essentially, in this case. So these are the three they have to know. And then it's activated by citrate, an upstream precursor. So remember the transport, right? How if we have a lot of citrate buildup, the Krebs cycle is going to slow. We have a lot of energy, and it's one way that we can stimulate building up fatty acid oxidation, because it's almost like a signal molecule for, hey, we have a lot of energy. The Krebs cycle is being backed up. And so why regulate the step? It's a committed step, as I mentioned, and the step requires energy because, again, biotin, it uses ATP. I want to emphasize that as well. It uses ATP every time. And so because it requires energy, it's very costly, and we don't want to just conduct this as many times as we want. We want this to be tightly regulated. So now talking about fatty acid synthesis, it's a 4-step sequence. It's going to look very similar to beta-oxidation, just in the reverse direction. So the enzyme that catalyzes this whole thing is called fatty acid synthase, and it has 7 parts. It's very complex. The only parts you need to know are the KS subunit and the ACP subunit. So the KS subunit is going to be loaded with acetyl-CoA, and the ACP unit is going to be loaded with the malonyl-CoA. And this is just the start of it, right? We're talking about like the first 2 building blocks. We're not talking about the intermediate of the fatty acid chain. So the first step that's going to happen is our condensation reaction, where we now combine the 2 building blocks together. That's condensation. So we combine the acetyl-CoA and the malonyl-CoA, and we lose the carbon dioxide in this process. So that's where instead of forming 5, because it's 3 plus 2, instead of forming 5, we end up forming 4. So as you can see here, the carbon dioxide leaves, and we have 1, 2, 3, and 4. And I just want to emphasize our malonyl-CoA is going to be the carbons that are on the first and the alpha carbon. The acetyl-CoA building block is going to be our beta, gamma, and then so on and so forth. So essentially, you can think of our malonyl-CoA being the foundation of the fatty acid chain, and the acetyl-CoAs stacking on one side. And that's the first step, right? So the second step is we're going to reduce this intermediate using NADPH. Remember, the shuttle generates the cytosolic NADPH right here on this bottom half. So this is very important to know. If you could be asked, like, where does the NADPH come from? It comes from that, or alternatively, the pentose 5-phosphate pathway, because we know it generates it from there as well. So something to know is like, okay, where can the NADPH come from? And same thing with the rest of these other molecules. Where could these other ones come from in the cell? It's a very good thing to know, and a good question to keep in mind. And if you can try this experimental. I haven't tried it myself, but like go on ChatGPT, ask it, hey, where are the possible sources of NADPH in a regular eukaryotic cell, in an animal cell? You can just go on to that, and then it'll give you a list of it. And then obviously the ones that make sense, good. But I haven't tested it, so this will be purely experimental. I'm not like advocating for that. But that's the second step. So the third step is going more in depth, what exactly got reduced, right? So we have our carbonyl group on the beta carbon, right? What's going to happen is instead of it being oxidized, right, it's going to be reduced into a hydroxyl group. So this looks very similar to like beta hydroxybutyrate. It looks very similar. And the thing is like, I'm pretty sure this is the same molecule. But I want to emphasize is that instead of going the oxidation direction, like in beta oxidation, we're going in the reduction direction. So that's the conclusion of step 2. And what ends up happening in step 3 is we're going to dehydrate this molecule. So that OH and the H from the adjacent carbon are going to like come together to form the water, and that's going to leave. And we're going to have this double bond form in place. And again, this is like the opposite direction of beta oxidation. Now the last step, and now that we have this double bond, the last step is going to be reduction. And so what ends up happening, again, we have this double bond. We're going to reduce it to form our single bond between the alpha and beta carbon. And that's exactly what we have here. And that's our chain right here, our 4-carbon chain. Now we add another acetyl-CoA onto it as well. And the other acetyl-CoA is going to be attached, again, to the KS group right here. Whereas the ACP subunit is going to retain that growing chain. So that's something to keep in mind. The ACP is like the growing chain, and then the KS is like bringing the incoming fatty acid monomers into there, and so they can attach themselves. Something to keep in mind. Oh, I lied. I'm so sorry. Before I forget. The growing chain is moved to the KS. My apologies. Yes, this makes sense. Yeah, and the ACP is reloaded with malonyl-CoA. Yeah, and you guys can walk through this as well. But like I said, on the last step right here, our 4-carbon chain that we just finished the first fatty acid synthesis 4-step process, this is going to be transferred from the ACP onto the KS. And so this is just another thing to go through. Again, you start from this right here where we have the ACP and KS is loaded with malonyl and acetyl-CoA respectively. Again, it's on this other slide too, so you can go through this and take your time with it, And yeah, so, and then let's say we built up our molecule, right? And now we have our palmitate, which is our 16 carbon fatty acid chain. Wow, I was literally blinking. So the way we end it is that the cycle repeats, and then until you form your palmitate. Now this requires 7 cycles because again we combine 2 of the acetyl-CoAs, and then we do 7 rounds of addition of the 2. If that makes sense. Again, just if you're not convinced, just do the whole process yourself one time, and then it'll become extremely clear like that. And so the possible fates are for the fatty acid synthesis is that it can be directly used in a different part of the cell. It can lengthen the chain for longer fatty acids if possible, and then it also can undergo desaturation, where we have double bonds and then we get rid of those double bonds as well. I don't know. Those are the 3 possible fates. That will be just a memorization thing. I completely forgot that. But essentially, yeah, the palmitate will just come off of the KS or ACP the KS subunit, and we can use it for these 3 different things. And this is just the energetics of it. We take 8 acetyl-CoA, 7 ATPs, 14 NADPHs to form our palmitate, 8 CoA, and then our respective oxidized or less energetic molecules. And so now talking more about how the beta-oxidation and fatty acid synthesis are regulated, they're opposing and they have to be coordinated so that only one occurs at a time, right? It's very similar to gluconeogenesis and glycolysis. Remember PFK2, PFK1, FBBase1, FBBase2? It's very similar. So there coordinates through malonyl-CoA. The malonyl-CoA can react with these 2 different pathways, and basically it'll stimulate fatty acid synthesis because it's the substrate. It's going to obviously want to stimulate it if there's a lot of the malonyl-CoA. And then it blocks beta-oxidation because if we have a lot of malonyl-CoA, then we want to build it up because we have a lot of energy, right? So that's the first step. But there are other things too in this diagram where this left right here is talking about fatty acid synthesis in the cytosol. So you can see this is like a cleverly drawn mitochondrion for the beta-oxidation. But here we see that glucagon stimulates our activation of... glucagon stimulates the PK, which will inactivate ACC, which again, it's fatty acid synthase, and that fatty acid synthase, acetyl-CoA carboxylase. That's what I meant to say. So as you can see here, our acetyl-CoA carboxylase is constitutively active in conditions where there's high insulin, there's high glucose, high energy, and we need to form fatty acid synthesis. But if we have glucagon, it'll stimulate PK and it'll phosphorylate it, and that inactivates ACC. You should be able to rationalize just from a physiological perspective. Okay, glucagon means we have low glucose, and we want to form glucose in the blood. So we don't want to build up fatty acid. We want to build up glucose. So we want to inhibit it. So, just rationally, you can think of it. And then this is just the mechanism of action that I definitely should know as well. That's everything for here. And then moving on to more of these very niche things as tags, triacylglycerols, and then glycerophospholipids. So we'll talk about each one. So both of these 2 use the same precursor, which is phosphatidic acid. So phosphatidic acid is going to form our glycerol-like backbone, and it's made from LG3P, and it's usually converted from DHAP, essentially. So for triacylglycerol synthesis, we're going to first replace the phosphate group with an OH group. So right here, as you can see, this phosphate on the 3rd carbon, it's going to be dephosphorylated, and the OH group is going to take its place using phosphatidic acid phosphatase. That makes sense. And then also, it's going to replace the OH with the fatty acid. And as you can see here, we start forming our tag. And this process just occurs for all 3 of the heads, or for the 1, essentially. And then the glycerophospholipid synthesis... And then for the glycerophospholipid synthesis, we attach a polar head group to phosphate groups through one of the 2 methods, see top right. And so method 1 and method 2. So method 1 is the activation of the phosphatidic acid. So, and method 2 is we activate the head group. So let's talk about the first one. CTP activates phosphatidic acid to form CDP diacylglycerol. And so it's this thing right here, strategy 1, where we have our phosphatidic acid. We basically end up attaching a CTP from this thing, and it's going to form this complex CDP diacylglycerol. Now what ends up happening is the OH on the 1,2 diacylglycerol, so on this thing right here, the OH group on the head group acts as a nucleophile, and it'll attack and basically displace the phosphate. The other 2 phosphates are the CDP, and it'll take over, and now we have formed our glycerol phospholipid, and that's our lipid here. Our lipids up here, and then this is our glycerol part, head group. And then method 2 is CTP activates the head group first, And then the OH on the 1,2-diacylglycerol will then do the same thing, but it's just on the opposite side. That's essentially it. These are two different methods. We don't know, but there's just different sites and locations that you guys should definitely know. If they ask you, okay, what are the two strategies of synthesizing triacylglycerols or glycerophospholipids? Or, sorry, what are the two methods for synthesizing glycerophospholipids? And so some membrane lipids overview, you should recognize the building blocks to be prepared to identify and distinguish. So I'll give you some of the hints, but these are just more the main ones. We know glycerophospholipids, sphingolipids, more sphingolipids, galactolipids, archaeal ether lipids, and sterols. We'll go through each one, don't worry. But the classification name gives you a preview of what to look for. So phosphate groups on phospholipids, so look if there's an addition of a phosphate group, a sphingosphene backbone in sphingolipids, and we'll talk about that, what makes them special. And then for galactolipids, you guys don't really have to know this, but just know that it's a mono or disaccharide called galactose, usually. Or it's like a saccharide or a sulfur. If you ever see a sulfur, then you know it's a galactolipid. And then ether lipids are connected via an ether linkage, so a COC, and all of them are single bonds. And we'll go into depth, I'll show you. And then lastly, sterols, which is just the rings, obviously. So this is some practice for that, but I just want to, we'll go over these details in depth. So this is an ether lipid. Why? Why is this an ether lipid, Alex? So look for the glycerol backbone, right? It's the 3 carbons, basically. And in here, we see the 3 carbons that are attached to O's, right? But in this case, we see that this is attached to an O, and that's it. This is not a carboxyl group. This is just a simple ether. And that's exactly what an ether lipid is to the glycerol backbone, to the fatty acid chain, it's going to be attached via this ether bond, this ether linkage. And that's what we see right here on these 2 carbons. Now, sphingolipid, it's a special one. The best way to identify it is a carbon-carbon double bond. If you see a carbon-carbon double bond on the glycerol backbone, which is right here, carbon 1, carbon 2, and carbon 1, carbon 2, and carbon 3. If we see a double bond linkage, then we know it's a sphingolipid, right? And then additionally, if you see an amide linkage right here between the glycerol backbone and also the carbon right here, as you can see, then you know it's also a sphingolipid as well. And then the last one is a sterol, which I don't really need to go in depth. It's just a 4-ring. You know it's like a steroid. So going into the synthesis of sphingolipids. So as you can see, sphingosine is derived from serine and palmitoyl-CoA. So palmitoyl-CoA is going to combine with serine. It's going to form this beta-ketosphingolipid, and essentially it's going to be reduced into sphingolipid because right here is beta-keto. We turn the ketone group into a hydroxyl, so we get rid of the name, and it's now sphingolipid, as you can see here. It's on this top part, carbon 3. And then from there, we add a fatty acyl-CoA to form N-acylsphingolipid. And that's it. And that's basically how we do this whole synthesis. Yes, guys should know this as well. That's annoying. I know you probably are like, Alex, like there's already so much. It's just another thing you have to put onto the memorization table, or maybe memorization like stuff. So again, sphingosphines are not built onto the glycerol. They use NADPH and they attach to fatty acids to form N-acylsphingolipid. And in animals, N-acylsphingolipid is oxidized to ceramide and then converted to sphingomyelin by attaching a choline group right here. As you can see, this is what a choline looks like. And the reason why we bring this up is because, sphingolipids are important because our blood groups are determined by glycosphingolipids. That's what we can determine between type O, type A, type B, etc., type AB. And then also with the RH factors as well. But that makes it more complicated. And then cholesterol, we know that it's made from sterols, and we'll talk about sterol synthesis in the next lecture. But essentially, we see here that it's a steroid nucleus, right? And notice the rings. You see ring 1, ring 2, ring 3, and you notice that all of them have the 4 rings in them. And that's what makes it very easy to identify, is that a lot of these will have the 4 rings, A, B, C, D, A, B, C, D, A, B, C, D, and whatnot. And obviously, like, there's like dopamine, which is only like 2 rings, I'm pretty sure. I know that those are different. Estrogen, all that stuff is, or no, estradiol is 4. But like, serotonin, that's what I meant to say. Serotonin, dopamine, those ones only have one ring, but they are derived from cholesterol nonetheless, I think. Or tyrosine. I don't want to confuse anyone. And so I just want to go over very quickly is that, just like a brief overview of the next lecture, is that isoprenes are sterols and fat-soluble vitamins, and they are the building blocks for cholesterol and for our lipid-soluble vitamins. So as you can see here, isoprene is a 5-carbon unit, and if we bring a bunch of these 5 carbons, we can end up forming our cholesterol. And also, additionally, we can attach them to form our vitamin A, vitamin E, D2, K1, if you guys want to know this, it's a plus. It'll help you on the MCAT for sure. It'll help you maybe for this class, but I don't think they're gonna really test you on what's vitamin A, what's vitamin E. But they may ask you like, oh, like what's it derived from, and which it's just isoprenes. And yeah, here's another practice problem. You guys can go through this, and we'll move on to lecture 14. I'm gonna just stop it here because I do have to go to that one thing. I took a longer time than I expected.